Hello everyone, this is Jason from STEM Nerds. Thank you so much for being here. In today's video, we are going to be talking about Mendelian genetics. We're going to be talking about how traits are passed down from one generation to the next. I'm going to show you in a future video how to do Punnett squares and how to figure out probabilities. Thanks for being here. It's kind of hard to believe that so much of what we know about genetics and about how traits are passed down from one generation to the next came from this gentleman here, Mendel. He was a monk in the 1800s who did a lot of work, believe it or not, with pea plants, and he was trying to figure out how certain traits are passed down from one generation to the next. And he made all kinds of assumptions that we now know a lot of them for a lot of living things really aren't true, but he totally lucked out with the characteristics that he was looking at and the traits he was looking at. His assumptions wound up being true. And even though we know now that some of those things aren't necessarily always the case, it really worked out in his favor and helped us understand genetics in general. So what did Gregor Mendel do? Well, this is a pretty good summary. What he did is he was studying pea plants and he studied thousands of them. So he had a huge sample size over many, many years. And what he did is he looked at certain characteristics. And so the characteristics are the things that you see at the top. Things like stem length, flower color, pod shape and color, seed shape and color. And so these things are characteristics. And then you have the traits, which are how the characteristics are expressed. And so when it comes to stems, you've got the stem, which is the characteristic, and you got the trait. Is it tall or is it short? And when it comes to the flower, you can have the trait either be purple or white. When it comes to the pod, that characteristic, well, is that pod color yellow or green? That's the trait. Is it smooth or is it constricted, inflated or constricted? Again, those are traits. And so the traits are how the characteristics are expressed. Eye color would be a characteristic. Is it blue? Are the eyes blue or green or brown? That is the trait. And he wound up studying a number of these different characteristics, and we're going to go into a few of them just to help us understand genetics. So one of the things that he discovered for certain characteristics, sometimes one trait would be expressed over another one. And he recognized that certain traits were going to be what are known as dominant, and other traits would be recessive. Another thing that he recognized is that living things are often getting two copies of a gene. A gene is a coding region of the genome, and you can have these two different copies, otherwise known as alleles. An allele is a version of a gene. And so here we're looking at this picture. Homozygous means that the organism has two of the same alleles. Now, it can be homozygous dominant, and that's usually represented by a capital letter. And so that's the capital B on the far left-hand side, in which case the organism has two alleles that, when present, will be expressed. You can have homozygous recessive. That is represented there by the two lowercase b's. An organism can also be heterozygous. That means they have a copy of each version of a gene or each allele. That would be represented by a capital B and a lowercase b. So let's take a closer look at how this plays out and exactly what Mendel did. And so as he was working with his pea plants, and this happens to be roses, but it's the same general concept, what he was doing is he was working with the parental generation. And that parental generation, what was known as true breeding. That means if you self-pollinated these organisms, they would always give rise to the same trait. And so in this particular case, you have red flowers, and if you self-fertilize the red flowers, they were always going to have more red flowers. And on the bottom, if you had white flowers and you self-fertilize those, they would always give rise to white flowers. And so that was called true breeding. But what he noticed is when you combine those two and you hybridize them and you wind up with basically offspring, you wind up with all red flowers. And he was trying to figure out why, and that's when he realized that that red flower, that red allele, is going to be dominant over the recessive. And this Punnett square, a Punnett square is a way to determine whether or not a genotype, the genetic makeup of an organism, is going to lead to a certain phenotype, the physical characteristics that we can actually see or measure. And so what we have here is we have a genotype at the top, which is capital R, capital R. And so that is a homozygous dominant organism. That red color is dominant. And at the bottom, we have homozygous recessive. That is the genotype of the flower at the bottom. Lowercase r, lowercase r. And so you can put these in a Punnett square. And I'm going to show you how to do this in another video, but you can put these in a Punnett square and basically come up with the genotype of the offspring and the related phenotype, physical characteristic. And so what we have here when we do this is basically what he noticed is every single offspring that came out red. Well, why? Because they all wound up being heterozygous. And again, I will show you how that happens later, but basically in the upper left-hand corner of that square, that flower is going to have one capital R and one lowercase r. Well, if that's the case, then that capital R is going to be the allele that's expressed. You're going to have a red flower as the phenotype. But the same thing is happening with the other three flowers. They're all getting one capital R and one lowercase r, and that gives rise to red. And so he was able to determine when something like that happens, well, the physical characteristic that you're looking at, that particular phenotype, red color, is dominant over white. And so basically in this picture, we have the exact same thing going on. The very, very top is what we just explained. They're using different letters here. All right, the letters really aren't all that relevant. I always recommend if you're going to use letters to represent a dominant and recessive allele, use letters that are easy to distinguish. The R in the previous slide, B here, pretty easy to distinguish. And so here we've got our true breeding parental strain at the very top. Capital B for our, what should be a red flower, lowercase b with another lowercase b, homozygous recessive for the white flower. When you cross those things, you create what's known as an F1 generation, filial 1 generation. And so you've got your parent strain that gives rise to F1. And the F1s, as we saw in the previous slide, they were all heterozygous. And so they are all one dominant allele, one recessive allele. And in this case, instead of being capital R, lowercase r, it's just capital B, lowercase b. It means the same thing. But here's what Mendel realized. When you start crossing those organisms, that F1 generation, so you have two heterozygous organisms that cross, look at the Punnett square here. It's showing you the genotype of each potential offspring. Now, this is not an exact ability, it's a probability of what's happening. But because Mendel did thousands of experiments, he always seemed to get the same thing. The phenotype, the physical characteristics, always seemed to be 3 to 1, right? So you had 3 red flowers to 1 white flower. And the reason for that is when you look at the genotype, well, you would wind up with the probability of having one homozygous dominant in the light blue. You would have 2 heterozygous, but they're both showing for red flowers. And so you have that 3 red flowers. But then there's a 25% chance that you would have 2 lowercase b's in this particular case. In other words, homozygous recessive, and that would account for around 25% of the phenotype. And so you can see here, you've got a ratio of 3 to 1, in this case, pink to white or red to white. And then you can see here in the genotype for the F2 generation, that is the generation that was born from F1, you've got the genotypic ratios, well, 25% probability of having homozygous dominant, 50% having heterozygous, and then 25% homozygous recessive. Now, as I've already said, Mendel made a lot of assumptions when he did his work back in the 1800s, and they were fair assumptions back then. We know things are a little bit more complicated now because we've had an extra 150 years or so to do some research. But there were 2 basic laws that he developed from his work, and one was, as you can see here at the bottom of the screen, the law of segregation, which simply says that particular law focuses on how one gene's 2 alleles separate, and so they're going to separate from one another. You've got one allele from the male, one allele from the female, and through meiosis, they're going to separate individually. The law of independent assortment is a little bit different. That says that each gene will separate from one another independently. And so in other words, a gene for flower color is going to have it's going to separate completely from stem length or pod color or pod shape. Now again, that was what he assumed back then. We're learning that that's not always the case, but luckily for him and for us, it was the case when he was working with these characteristics for these peas. And so basically the law of independent assortment looks kind of like this. We've talked about both mitosis and meiosis in previous videos. Check those out if you haven't seen them, because I think they'd be really useful for understanding this. But when you have the law of independent assortment, what it's saying at the bottom, so you've got prophase 1, but in metaphase, you can see that the homologous pairs are lined up in either way. It doesn't matter, as long as each new cell is getting a full complement of genetic material. That's all that matters. It doesn't have to be all parental DNA on one side, maternal DNA on the other. It can be mixed as long as each new cell, like I said, has a full complement of DNA. There were some other assumptions that he made and things that made life really easy for him. As we saw in the previous example, when you had a red and a white flower and they were hybridized, you would get either red or white. But there are instances where you don't get either red or white. You get a blending between the two, and that is called incomplete dominance, when both of the traits are mixed together and you wind up with an intermediate. And so instead of a red and white for your heterozygous organisms, when you have a characteristic that is going to be showing incomplete dominance, you get a mix. And so in this particular case, you'd wind up with pink flowers. Again, with his particular experiments with the peas, the dominant allele was always fully expressed over the recessive allele when it was present. But that doesn't always happen. I think we know that intuitively, even with human beings. If you have somebody with black hair and somebody with blonde hair and they have children, it's not going to either be black hair or blonde hair. Usually it's some shade of brown. Same thing with skin color. If you have somebody who's very dark skinned and they have a child with somebody who's very light skinned, usually it's not going to be someone that's either very dark or very light. There's going to be a mix between the two, and that's incomplete dominance. What Mendel was doing with his peas is he was working with characteristics that showed complete dominance. In other words, again, as I said, that dominant allele was always expressed over the recessive allele if the dominant allele was present. Something else that can often happen, and that's not dominance or incomplete dominance, it's another thing called codominance, where you can have two traits for a characteristic that are fully expressed, but in different parts of the animal. And so here we've got an example of a cow. You've got a brown cow and a white cow, and they mate, and you wound up with a brown cow with white spots or a white cow with brown spots. That is an example of codominance. So in codominance, you have both of the traits are fully expressed, but in different parts of the body. Something else he got really lucky on, and that is the characteristics he was looking at really only involved one gene, and we know now that that is absolutely not the case for the vast majority of genes, certainly that are comprised in the human genome. It's way, way more complicated than that. We don't have, unfortunately, a gene for strength or a gene for speed or a single gene for intelligence or body mass or obesity. There's lots and lots of potential gene interactions that can lead to physical characteristics, certainly in humans and in most other organisms as well. And so these are three concepts at the top of this table that I think we should understand, and that is what you can have a very messy situation. So the first one is epistasis. In epistasis, what you have are multiple genes that are interacting. And as you scroll down or as you go down, you'll see that that effect is that one gene may mask or modify another one. The really good example is a Labrador Retriever. That's a type of dog, and I think most of us know that there are black labs and chocolate labs and white labs. Well, it turns out that there is a gene that allows the dog to express a color in their coat, black or brown. But there's another gene that determines whether or not that gene is ever going to be expressed, and that's where we get the white lab from, because that second gene blocks the gene for the characteristic of coat color in and of itself. So it's not three genes that are interacting. It's actually two, one that expresses either black or brown, and a second gene that determines if that gene can be expressed at all, and if it's not, it'll lead to a white lab. Polygenic inheritance is another situation that can happen where it's not just a single gene that is having an impact. Here, what we have are many genes that contribute to a single trait. And this is what I was talking about before, where we're talking about human height or weight or skin color or strength or intelligence. There's not just one gene that controls those things. There could be dozens, maybe hundreds of genes that control those physical characteristics. Now, pleiotropy is an interesting one because that is one gene, but that one gene doesn't impact one single characteristic that leads to one trait. It's actually one gene that has an effect on multiple phenotypic characteristics. Sickle cell anemia would be a really good example of that. So if you think about sickle cell anemia, what happens there is there's a single mutation, and that single mutation causes a red blood cell to lose its shape, going from kind of almost like a circular donut shape into more of a crescent half moon sort of shape. And when that happens, that leads to lots of different physical characteristic changes, right? So a lot of phenotypic changes. You have people who get anemia and they're prone to blood clots. Now they are less prone to malaria because the malarial parasite can't infect that sickle cell gene as easily as other types of genes, as easily as other types of cells. And so pleiotropy, one gene having an impact that leads to multiple phenotypic effects. It's kind of interesting knowing that because you can actually use this in real life. Now here we've got an example of flowers, and what we're trying to do here is something called a test cross. And so in this particular example, let's say you've got some purple flowers, and I know it's going to be a silly example, but let's say you are a flower breeder. You grow flowers, and these purple flowers are worth a fortune. Unfortunately, the white flowers are nearly worthless. They're really common. And what you want to determine is you happen to have some purple flowers, but you don't know why they're purple. Are they purple because they are homozygous dominant? The purple is the dominant allele. Or are they heterozygous? You don't know why at this point. You know the phenotype, purple. You don't know the genotype. And so what you can do is you can cross that purple flower with a flower that is homozygous recessive. Now I wish this was a better example in terms of the letters that they use, because I just got through saying, and this is a really good example of why, when you are picking letters to express dominant and recessive alleles, pick letters that are different. R is a really good one. You saw B, that's a great one. The letter A. The letter P, not a really great option. If you were using the letter O or Z, also really bad options. But here in this example, I can't change it. They used P. So anyway, what we're doing here is we're trying to figure out, okay, for the purple strain, is this homozygous dominant or is it heterozygous? And so you set up at what's known as a test cross. And the test cross, the purpose is to figure out what is the genotype of the organism expressing the dominant phenotype. So on the left-hand side, we have basically two options. So we have two Punnett squares set up here. On the left-hand side, option one is you have the recessive at the top, and then you have over there on the side, you have what would happen if the purple organism is homozygous dominant. Well, guess what? Capital P would always be expressed over lowercase, so purple always expressed over white, if the purple flower is homozygous dominant, all of your offspring would be heterozygous, and therefore they would show the dominant phenotype being purple. Go to the right-hand side, that right Punnett square. Now let's assume, okay, on the top, again, we've got our two lowercase p's, and that is our white flower, but what if our purple flower happens to be heterozygous? Well, what we're going to see in the offspring is around 50% of the offspring from that mating is going to show the recessive phenotype, the white flowers. And then you'll know, okay, my purple flower is heterozygous, it's not homozygous dominant. Now, you don't have to have just one characteristic to do a Punnett square. This is an example of what is known as a dihybrid cross, where you're actually looking at two different characteristics at the same time. And so in this particular example, what we have is we have the seed shape and also the color. So P shape and P color. And what we have to do in this particular case is we have to designate letters that represent color. And in this particular example, you could see capital Y is for yellow and lowercase y is for green. And so hopefully what we're already learning is that the lowercase y, that green color, is a recessive trait, a recessive allele. Then we have shape. We can see that the capital R is for a round seed and lowercase r is for a wrinkled seed. So once again, we should know already that it must mean that the round shape is dominant over the wrinkled shape. And so what happens is, let's say you take those true breeding organisms, and so you have homozygous dominant for both on the left-hand side and homozygous recessive for both on the right, the shriveled green pea, poor sad little thing, and you mate them. all of your offspring are going to wind up being heterozygous for both of the characteristics. Now we look at the Punnett square. It gets a whole lot messier. And again, I promise you, I'm going to do another video right after this. And then we're going to do this on the whiteboard. And I'm going to show you how to set these up so that you are an expert, so you know how to do both mono and dihybrid crosses, and later on, even sex-linked traits. But you can see this F2 generation. There's a lot going on here. There's a lot of potential combinations. And for now, what I want us to see is the very bottom. We have what is known as the 9:3 to 3:1 ratio. And this is always the case if you have two heterozygous organisms that are heterozygous for both characteristics. What's happening here is you get the same ratio pretty much all the time. And what it means is 9 out of 16 potential genotypic combinations are going to result in an organism that has dominant phenotype for both. So yellow and smooth. Roughly 3 out of 16 of your potential combinations are going to result in something that is a smooth P, but it's green. And so it's going to have one dominant phenotype and one recessive. The other 3 out of 16 are going to have one dominant and one recessive phenotype, but they're switched. And so now we have a wrinkled yellow. And 1 out of 16, you have a 1 out of 16 probability of having homozygous recessive for both of the characteristics. So you're going to have a green P that is wrinkled. It's important to note that when we're doing this, when we're doing these Punnett squares, what we're trying to do is we're trying to come up with a probability of what we're going to see in offspring, but it's a probability, not an exact ability. Nature sometimes throws a wrinkle in things, but this is a great way at least to determine probabilities, especially if you know what genes, the genotype of an organism for a particular phenotype. And this is just another example right here of the exact same thing we looked at, but it's a little bit easier to see all the different combinations. And what we're going to be doing when I draw out the combinations on the whiteboard is I'm going to show you, okay, where are these letters coming from? How are we getting these combinations? But hopefully we can kind of see the different combinations on the top and the sides. But like I said, that second video that we do, we're going to really go through this. And one of the cool things is once you understand how genes are passed down from one generation to the next, you can do something called a pedigree. And that's what this is right here. And what a pedigree is doing is it's allowing us to see on a family tree which individuals carry certain alleles for certain genes and which ones don't, and the probability that they were passed down, and why we see certain phenotypes throughout a particular family. We're going to get to this. I'm going to give you an example in the next video where we actually kind of figure out a phenotype or figure out certain phenotypes and genotypes based on some information we learned from some offspring and work our way back to the parental strain. All right, everyone, so that was a quick rundown of Mendelian genetics and how Punnett squares work and the differences between phenotypes and genotypes. What we're going to do in the next video, super important to watch, we're going to actually do some Punnett squares. I'm going to show you how to set them up. I'm going to show you how to work through them. As always, thank you so much for being here. Have a great week, everyone.